Hi class, let's get started on our discussion of bone tissues and bone growth. We've already talked about compact bone tissue and spongy bone tissue in a previous recording. In this recording, we're going to talk about the marrows and bone development in particular. As we look at our bone marrow, there's two kinds of bone marrow in our body. We have red bone marrow and yellow bone marrow. And I'm going to go ahead and pull up the laser pointer. Here we go. So as we look at this red bone marrow is a myeloid tissue. When we say myeloid, it's going to be producing blood cells. It's going to produce all the blood cells. We have our red blood cells, white blood cells, and our give me a moment here. Our thrombocytes, our platelets. I almost said clotting blood cells, and that would have been an incorrect term. So our platelets. Now, as we look at red bone marrow, this figure right here shows an adult skeleton. In an adult skeleton, we have red bone marrow in the spongy bone tissue of the axial skeleton. As a child, though, we're going to have red bone marrow in nearly every bone of the body. In particular, if we look at the medullary cavities of long bones, in children, those medullary cavities are filled with red bone marrow. As we age, that red bone marrow is going to be displaced by adipocytes or fat, also known as yellow bone marrow. This yellow bone marrow is going to fill the medullary cavities of our long bones. In the instance where we have chronic anemia or chronic low red blood cell counts, we can have the option of taking this yellow bone marrow and reforming it back into red bone marrow, but that's fairly unlikely. In terms of our bone development, our bones are going to go through several stages of development. As we look at this process, this development of the bone is known as ossification, sometimes known as mineralization. At times it's been referred to as osteo meaning bone, genesis meaning creation. So osteogenesis is creation of bone. And as we look at this creation of the bone, there are two methods that our bodies use to create bone. We have intramembranous ossification, where we're going to have two separate membranes, and bone will form in between these fibrous membranes. This is typically a method that's going to be used to make flat bone. So when we think of flat bone formation, think of intramembranous ossification. Intramembranous ossification is not the most common kind of ossification. Endochondral ossification. Chondral meaning cartilage, endo meaning inside. Endochondral ossification is the process of turning cartilage into bone. And that is by far the most common kind of ossification. So as we're looking at intramembranous ossification, we start first with, in step one, our membrane. We have a superior and then a deep membrane. And within these membranes, we're going to have some osteoid tissue forming, some zones of ossification. And then during step two, that ossification tissue or osteoid tissue is going to continue to expand. We'll have osteocytes develop out of the mesenchymal cells. Those osteocytes and osteoblasts are going to excrete calcium and phosphorus and other minerals into the extracellular matrix to continue to ossify the bone. As we look at step three, that flat bone is going to be overwhelmingly spongy bone. There'll be the fibrous membrane on the surface. So we'll have our periosteum, and then a whole bunch of spongy bone. But spongy bone, just a periosteum, is not very strong. So we're going to take this periosteum, or the spongy bone, and reform some of it into compact bone. So we'll have kind of a compact bone sandwich. We'll have compact bone, spongy bone, and then more compact bone, making our flat bone tissue. Here's a photomicrograph showing the same thing. So we can have our fibrous layer, our periosteum, followed by our osteogenic layer, and then here we have the formation of spongy bone. This spongy bone is going to have osteocytes embedded within it. Osteoblasts are going to form the perimeter, both the superficial and the deep perimeters. And then we're going to have more fibrous tissues on the inferior deep margin. The most common form of ossification is endochondral ossification. The general pattern here is that we start with cartilage to bone. So cartilage turns into bone. So here we have our early cartilage pro-runner and their prototype. And this prototype bone that's shown here is going to enlarge. We'll have more and more chondroblast expanding this cartilage framework. And as this is growing, we'll have a zone of ossification, or excuse me, a ring of ossification around the perimeter. We'll have the periosteum serve as an ossification center. In the very center of that cartilage, we'll have another ossification center form. We'll have, in step 2, penetration by the vasculature, so that we'll have blood vessels penetrating to form the precursor of the marrow or medullary cavities. And we're going to have more ossification occurring on the inside of the bone now. We'll also have secondary ossification centers form at the epiphyses, both the superior, or excuse me, both the proximal and the distal epiphyses. And I'm not really sure which is proximal, which is distal, based on just looking at this bone right here. For those of you who are sticklers on directional terminology, if I look over here, we definitely have a proximal and definitely have a distal humerus, phalange on this end. So here, backing up here, we are now going to have our medullary cavity form within the diaphysis, and then within our epiphyses, we have secondary ossification centers. Left over in the middle, we're going to have some hyaline cartilage. This hyaline cartilage that's left over in the middle is referred to as the epiphyseal plate. And that epiphyseal plate is going to be present as the child is born and as the child is prepubescent and pubescent. And that epiphyseal plate will form an epiphyseal line, a line of compact bone tissue, once that child is in their late teens, or I should say once that young adult is in their late teens and or early 20s, and they are going to stop elongating that long bone. So those epiphyses that are present, just to back up a little bit, so the epiphyses, so either the proximal or the distal epiphyses, are going to fill with spongy bone. That spongy bone is going to potentially be a site of red bone marrow for the adult, depending on their metabolic needs. The cartilage that's in the bone within it can be left over to form the epiphyseal line. We'll also have cartilage left over on the very distant ends. That cartilage that's left over is referred to as the articular cartilage. This articular cartilage serves two purposes. It is going to be a source of cushioning, and primarily it's going to reduce friction so that that joint can be smoothly moving, or that articulation point can freely articulate. As we're looking at this process of having the cartilage turn into bones, this endochondral ossification, those epiphyses, those gaps in between the epiphyses, those epiphyseal lines, are going to eventually fuse to form epiphyseal plates. So here we can see a fetal x-ray, or a newborn infant's x-ray, a young child's x-ray, and notice how there are lots of gaps in between the bones. There's not that much, particularly focus on the hands right here, there's not that much bone tissue or osseous tissue in the hand yet. As the child ages and becomes a young adult, the epiphyseal plates, which you can see here in these x-rays, fuse to form epiphyseal lines, and then the bone no longer grows long, increases in length. And that's all we have on our marrow tissues and bone development. I hope you enjoy this recording. Happy studies!